Great. So we're excited to have Brian Rich with us tonight, coming from Virginia, the Tick Lab at Old Dominion University. Brian has a degree in biology from UTRGV, a master's in entomology from Texas A&M. He's currently doing his PhD at Old Dominion University, working in the Tick Lab, and he is going to talk to us tonight about ticks of South Texas. So hopefully we've got all the technology set up so that we can hear him. And Brian, I'm going to turn it over to you.
Can you hear me okay?
Yes.
All right. Well, howdy. Thanks for having me. Always enjoy reaching out to the community to talk about my favorite subject, which is ticks. So let's talk about some ticks of South Texas. So ticks are arachnids. They're in class Arachnida, along with spiders, scorpions, vinegaroons, opolonids, mites. Their characteristics: they all have 8 legs, 2 main body segments. They do not possess antennae. They all have mouth parts that are called chelicerae. So if you're curious as to what that is, just think of the end of a scorpion, those little pincer things. The arachnids all have those or some sort of analog to it. So ticks are distantly related to the mites, though they are grouped with them. There's 3 main families. I'm only going to discuss 2, since the third one only exists for a single species in Africa, and they're weird. So the first family is family Exodidae. So these are the more familiar ticks. We call them hard ticks. So they all have mouth parts that you can view from above and below. They have a scutum or dorsal shield that's present, and this is why we call them hard ticks. You have that hard shield. And this differs among sexes. They have palps, which are right next to their mouth parts that they use to feel around and taste with. They have 3 obvious segments. And they breathe through spiracles, these little plates that sit behind their 4th pair of legs. So ticks, as the hard ticks, have several life stages. They start out emerging from the egg as a 3-legged larva. They'll take a meal, a single blood meal, molt into an 8-legged nymph. They will then take another blood meal, molt into their final stage, which is an adult, either a male or female, take one last blood meal, mate, and then start the whole cycle over again by laying a bunch of eggs. So hard ticks can only exist on a single food source. They need blood meals. They are obligate blood feeders. So they have a wide range of different strategies in order to obtain that blood meal. Some of the more common host-seeking strategies are questing, ambushing, and nesting. So when ticks quest, the ticks will crawl up onto tall grass and wait. And they're waiting, hoping that a host will brush by them, and then they can grab onto that host, climb up, taste it with their palps and see if it's a good host. And if it is, climb up, find an attachment site so they can feed. Ambushing is used by more aggressive and larger ticks. This is where they'll wait under rocks, in sand, or under leaves, waiting for hosts to pass by. And as they pass by, they charge them at full speed, and ticks can move quite fast. If you've seen fast spiders, ticks can move just as fast. And they'll wait and climb up onto you and find an attachment point and feed. And then finally, there's nesting. So this is employed by a large number of ticks, and it's the easiest route. The best way to ensure that you can get your blood meal is to always be there with the host. So these are ticks that will live in caves, in burrows, dens, and in bird nests. They'll come up to feed, and when they're done, they'll go back down into the middle of that nesting material where they're away from pecking beaks and claws, molt into their next stage, and wash, rinse, and repeat. So some of you have probably heard of seed ticks. So this is the first stage of tick. This is the tick larva. So they have 3 legs. They're very, very small. They do not have their spirochelar plates. Since they're so small, they simply absorb oxygen through their cuticle. They're also the most vulnerable stage of ticks, as they don't have the full waxy coating that nymphs and adults have. So they're very susceptible to things like acaricides and especially to saturation deficit. Desiccation is the worst enemy of the tick. So here I have several images. The first up top on the right is of a tick larva. This is a hard tick larva. Down at the bottom left, I have placed this tick inside the O on the of a penny, just to show you how teeny tiny it is. Now, to ensure their survival, larval ticks will clump together in what are referred to as larva bombs. So on this canvas flag here, we can see that all the ticks have clumped together. Now, they will clump together like this up on grass, and when you walk by, they will all get up onto you. Not all of the ticks are going to make it, so this ensures that at least a few larvae can get their blood meal, get off their host, and molt to the next stage. So ticks have 2 main body segments. They've got the head, which is the capitulum, and then their body, where they have their legs, spirochelar plates. The first leg is very important on the tick because it possesses a specialized organ called Haller's organ. Now, this organ is able to detect the presence and concentration of carbon dioxide in the atmosphere. So this means that when a tick is sitting up on a blade of grass, your exhalation or the exhalation of any potential host can be picked up by the tick and activated. The ticks will get excited, they'll wave their front legs around, trying to figure out where that plume of carbon dioxide is coming from, so they can get ready to get up on you. Ticks do, in fact, have an anus. I've heard from many people I've spoken to that they do not, but they do. Ticks need blood, but they don't need the liquid in the blood. They only need that hard packed material. And once they've processed it, they'll excrete it out as guanine and heme. So some ticks do have eyes. Here we have Amblyomma tenellum. This is the imitator tick. And if you follow the insertion point of leg 2, that's the second leg from the top, into where it inserts in the body, you'll see a sort of semi-clear eye. Now, this is a very simple eye. It's theorized that they can see things like a little bit of movement, and they can detect shadows. And much like an insect ocelli, they're useful for determining how many hours of daylight it has. Since in order to complete the life cycle, some ticks need to know when time is running out so they can get a host before winter starts. And of course, as hard ticks, they have a hard scutum. Now on the larvae, the nymphs and the females will have a scutum much like you see on the left, where it's covering up only a portion of their body. Now the rest of that is called alloscutum. And this is tissue that's been selected by nature to expand outward like a big bag. So I'm sure you may have seen ticks that are on dogs and other animals, and they look big and gray. That's because that alloscutum has grown outward so that it can take up the maximum amount of blood. Now you notice that the male Well, he doesn't really have that. The male on the right, his entire backside is covered by that scutum. So the male doesn't need a whole lot of blood since he doesn't have to produce eggs. All he has to do is take up enough blood that he has the energy to go and find a female. Some one-host ticks will have males that will bite in, and unlike the other ticks where they only take a single blood meal per stage, they'll actually take a blood meal, find a female, mate, then dig right in and get another blood meal so he can go mate again. So here we see Howler's organ on the front leg. It's in this little pit that you see in the bottom right. So here's a SEM or scanning electron microscope image I took of what Howler's organ actually looks like on a tick. And it's filled with a wide range of sensory cells and seta that can feed that tick all kinds of information. Heat, where that carbon dioxide is coming from. There's chemoreceptor cells in there so they can detect volatiles on your skin. So here now we're looking at the head of the tick, the basis capituli. So this is the business end of the tick. There's the palps, which are useful for tasting, pressure sensation, figuring out, is this a good host or not? Now, on the image on the left, we can see that the palps are covering the chela, or the cheliceraal bases. Now, these are analogous to a spider's fangs. And at the tips of these are little razor blades, and they flick out and they cut a hole in the host so they can insert its hypostome. And if you look at the image on the right, you can see that toothed hypostome surrounded by the palps. So the hypostome is covered in recurved denticles. Once the hypostome is anchored into the host, these denticles make it very difficult to remove. In addition, some of the salivary compounds that the tick has are used as a form of superglue. So if you've ever pulled off a tick on your dog, you'll notice there might be a little plug at the end of it. Well, that's tissue that you pulled out that was glued in place by that superglue. So here are the chelicera. So these are the cutting elements, and they sit on those two stalks that are covering the top of the tick's mouth. And we see they've got these fine razor claws so that it can slice open a nice hole in which to insert that hypostome. They are so fine that the host does not feel a thing. In addition, the salivary proteins that the tick is pumping into the host act as analgesics. So they cut off sensation from your nerves so you don't feel anything. Now, this is important because unlike a mosquito, which will land, feed, and get gone, our ticks need to feed for multiple days. So this involves biting in, secreting enough saliva with proteins that defeat your immune system so that they can keep that feeding lesion open and ensure sufficient blood flow so they can uptake a sufficient meal to molt to the next stage or mate or lay eggs. So we sort ticks in a variety of different ways. So here we have 3 different genera of ticks, and they all look a little different. Now, these are all male ticks, but if they were female ticks, their scutum would probably be slightly different from each other. In addition, their palps are different, and the lengths of their mouth parts are different. So these are important features that we have to figure out which genera and which species we're dealing with. which is important because different species can vector different pathogens. These are the sporactor plates, and the little dots inside are goblet cells. This is how the tick breathes. So much like insects, there is a passive oxygen uptake with tubules going to all over the body to oxygenate the cells. Now, unlike in a lot of insects, they can actually close off these plates and survive without oxygen uptake. In fact, here at the Old Dominion lab, they experimented with how long a tick could live in, say, an aqueous environment. So they sealed up some ticks inside a bottle of water where they had taken the oxygen out. It was just only water. And they found that the ticks survived up to 70 days. Now, this can vary from tick to tick, but that's a long time where they can live and survive flooding. You also notice that the sporactor plates are different in shape and size. And depending on which genera you're looking at, this is another way to figure out which species you're dealing with. We also identify ticks by their spurs. So if you turned a tick over and looked at it on its ventral side, you're going to notice that there's probably several spurs coming down from the bases of the legs. Now, these are useful for the tick in staying inside a hair coat on an animal while it's feeding. But different species will have different numbers of spurs and different size spurs. So here I have the spurs on the cayenne tick as opposed to the single spur on the Gulf Coast tick. So I've mentioned one-host ticks. There are three-host ticks or multi-host ticks. There's the most common life cycle. And then we have one-host ticks. So a one-host tick will get up on one host and it will stay there. It'll get up on that host as a larva, feed, back out of its exoskeleton when it molts, bite in immediately for a few hours or maybe a day after its cuticle is hardened, and repeat that cycle until it's an adult and mated, and will only drop from the host as an engorged female ready to produce eggs. Multi-host ticks, or three-host ticks, will get up on one host, feed, and when they're ready to molt into the next stage, will drop from that host, crawl off into the environment where they can safely digest their meal, molt to the next stage, and then either ambush or quest for a new host. Now, this new host may be the same species that it had before, or maybe something entirely different. Now, this is important because different species have different pathogens associated with them. So while a one-host tick will have to pick up pathogens from just that one host, and that one host pool of pathogens associated with it, a multi-host tick may be exposed to pathogens from several different pools, from several different hosts. So one of the most common ticks we have in South Texas is Ixodes scapularis. This is the black-legged tick, or the deer tick. It's famous as it is the vector of the causative agent for Lyme disease, a bacteria called Borrelia burgdorferi. It'll also vector anaplasmosis, in particular Anaplasma phagocytophilum and Anaplasma marginale, and also a pyroplasm, Babesia microti. So this particular tick goes through its life cycle in 2 years. So the larvae and nymphs will feed on small animals, rodents, rabbits, In California, they go after fence lizards. In Texas and along the East Coast, they prefer things like white-footed mice, which is a maintenance host for Borrelia burgdorferi. They'll also get up on dogs and cattle, horses, but that's primarily as adults. So in the southern parts of the US, the immature ticks become active in the spring and summer, while adult activity spans autumn to spring. And this can change depending on which part of the country you're in. So while this is true for Texas, here in Virginia, because we have such a stronger winter, the tick activity is limited a little bit more towards the fall. So this is the other ubiquitous tick in South Texas. This is the Lone Star tick, Amblyomma americanum. It gets its name from the female, you can see on the left, who has that small iridescent dot at the bottom of her scutum. So Amblyomma have eyes, long mouth parts. The male does not have any spots, but does have sometimes has little streaks of iridescence along its margin. And the larvae and the nymphs are referred to as inornate, so they don't have any color to them. They just look brown. These ticks have a reddish look to them, or maybe a tan color. They're a vector of ehrlichiosis, which can occur in humans and animals, especially Ehrlichia canis. And they are a known vector of Borrelia lone stari, which is the causative agent for southern tick-acquired rash illness. Lone stars are generalist feeders at all life stages. At all life stages, they will bite into people. Their preferred hosts, I believe, are small birds, small rodents, rats, possums. As adults, they will try and seek out larger prey, something like dogs, coyotes, horses, cattle. In particular note is that this tick has been demonstrated to afflict some people with the alpha-galactose meat allergy. So alpha-galactose is a sugar molecule found in most mammals. It's not found in fish, reptiles, birds, or people. Once you've gotten this allergy, it makes you allergic to red meat. So no pork, no beef, no rabbit. And this also includes gelatin, cow's milk, and milk products. So this is Amblyomma inornatum. These ticks are very small in size. They are a vector of spotted fever, so Rickettsia species, and also Ehrlichia chafensis. They're quite susceptible to saturation deficit. They prefer wet environments, so they'll often appear several weeks after a good rain. Their hosts include small mammals, deer, cattle, dogs, and of course they will bite humans. They are inornate, so they have no ornamentation on their scutum. They do have long mouth parts, and they are very, very small. This is Amblyomma maculatum. This is the Gulf Coast tick. It is a known vector of Rickettsia parkeri, which is the causative agent for spotted fever. It also can vector hepatozoon americanum. It's a causative agent that causes American canine hepatozoonosis in wild domestic dogs. They have an aggressive host-seeking behavior. So while the larvae and nymphs will quest, the adults may either choose to quest, or if they're running out of energy reserves, they may resort to ambushing. The larvae will feed on hispid cotton rats, birds. In Texas, they'll go after meadowlarks and quail, as well as turkey. There's increasing evidence that the nymphs may also attach to large animals, such as cattle. The adult ticks will attach and feed on cattle, horses, deer, sheep, feral swine, coyotes, dogs, cats, and other carnivores, and they will, of course, bite into people as well. So I'm including an image of the male and female imitator tick. I didn't have one for the cayenne tick, but there wasn't much point as only an expert can really tell them apart. They're almost identical, but their genes are quite different and they aren't really related that much. So we have Amblyomma mixtum and Amblyomma tonellum, which is the cayenne tick and the imitator tick. Now they have similar feeding habits. They both prefer the same host. They're generalists like Americanum is. At all life stages, they'll really attach to a wide variety of hosts, including people. Larvae and nymphs will feed on small things, rabbits, hispid cotton rats, quail, and the larger adults will feed on coyotes, javelina, cattle, horses, sheep, dogs, and of course humans. And they can vector various anaplasma species and spotted fever, rickettsia species. The American dog tick, Dermacentor variabilis. So these ticks are always found with ornation, which means that they don't have a plain scutum. So you can see here the female and the male both have some grayish or whitish streaks on their scutum. They're known vectors of the causative agent for Rocky Mountain spotted fever, Rickettsia rickettsii, which is my least favorite rickettsia. They can be found along the edges and trails of walkways. The larvae and nymphs prefer small mammals. Again, numerous rodent species, rabbits, cats, feral swine, dogs. The adults really seem to prefer canids, so foxes, dogs, coyotes, but they are known to feed on other things like cattle, raccoons, opossums, and of course, they will go after people if possible. This is the winter tick, Dermacentor albopictus. Now, these can be ornate or inornate. They're vectors of Rocky Mountain spotted fever, Rickettsia rickettsii, and also Anaplasma marginale. Their primary hosts include large ungulates like horses, mules, deer, elk, and importantly, moose. So if you've ever heard of ghost moose, this is the tick responsible for that. Winter tick adults are active during the winter, and this includes up north where it gets really cold and snowy. They'll get up on moose in such numbers that they cause severe irritation. And the moose, in response, in an attempt to alleviate this irritation, will vigorously rub up against objects in an attempt to dislodge the ticks. Now, this invariably results in loss of a lot of hair, which is where the ghost moose get their name from, since this leaves them with lots of whitish patches and also increases the likelihood that they're going to succumb to the elements. There's infrequent collections of them hosting on dogs, feral swine, and also humans. The brown dog tick, Rhiphocephalus sanguineus. Dr. Teal at A&M used to comment the brown dog tick because they only feed on brown dogs. Of course, they don't. They like to feed on everything. They're vectors of Ehrlichia, including the causative agent for human monocytic ehrlichiosis, or HME. Also, they're vectors of Rickettsia rickettsii, and there have been some recent outbreaks of this in New Mexico and Colorado. Dogs are the primary host for this tick across all stages in North America. This tick is also the most widespread. We bring dogs everywhere, so we have brought this tick everywhere. The larvae and the nymphs have been known to attach to other animals, small birds, rodents. All stages are occasionally known to bite humans. So once hatched, these ticks can persist in an environment for an abnormally long period of time. So nymphs and larvae can persist for anywhere from 6 to 9 months. And adult ticks have been reported to survive unfed for over 18 months. I have heard horror stories of people moving into a new home or new apartment where the previous owners or residents kept dogs, and their presence, their warm bodies and carbon dioxide, caused the ticks to become excited, causing mass amounts of ticks to start questing up the indoor walls, much to the alarm of the new occupants. So you guys live in a very interesting area. You live in an area where there is a historic battle against a historic tick. And those are the cattle fever ticks. Boophilus annulatus, the cattle tick, and Boophilus microplus, the southern cattle ticks. These are vectors of a couple different species of Babesia, Babesia microti, and Babesia bovis, the cattle fever and Texas cattle fever, respectively. The hosts are cattle, and they're primary hosts of cattle ticks. However, the species is also known to parasitize horses, sheep, goats, various wildlife, dogs, and cats. So I'm sure you've all seen these signs out there warning you that you are at the tick eradication quarantine line. So this is a stretch of land that comes from the mouth of the Rio Grande all the way up to Del Rio, Texas. And this is monitored by the USDA Animal Plant Health Inspection Service, along with the Texas Animal Health Commission. We really do not want this tick coming back into the United States. So the cattle fever tick eradication program took off in 1906 and has been running ever since. The ticks were declared officially eradicated in 1947. Growing up in Texas, I learned about the cattle trails and the Chisholm Trail and the Goodnight Loving Trail, and Texas cattlemen taking their cattle up to market to the Intercontinental Railroad to get that meat headed all the way out to the major city centers on the east and west coast. Well, there's a reason why there's so many of those trails. It wasn't just that some trails were being overused by lots of different ranches. It's the fact that the Texas cattle brought this tick with them wherever they went. So when the cattlemen would take their cattle up to the trails, well, the northern cattle ranchers would notice that their cattle tend to die off about the same time. It must be something with those darn Texas cattlemen. And what it was Texas cattle fever being vectored by this tick. And eventually, instead of greeting these Texas cattlemen with waves and hellos, they started greeting them with shotguns. So the Texas cattlemen had to move over from one trail to another, and another, until the Bureau of Animal Industry figured out that, well, it's this tick that's responsible. And they set up this program in order to get rid of it. So in 1906, this is the range of the cattle fever tick on the left, all the way up into California through Texas and much of the southeast. On the right, in the blue, we can see where that tick is today. It is endemic in the northern part of South America, in Mexico, and throughout Central America and the Caribbean. So I haven't talked about that other family of ticks yet. And that other family is called Argacidae. Now, these are the soft ticks. So unlike the exotids, they don't have that shield on top of them. They're all soft and kind of vinily or leathery. They look like little mean beans. So their capitulum's on the underside of the body. So if you look at them straight on down, you're not going to see their mouth parts. They do not have that dorsal shield. Their palps are small and spindly, and they've got 4 segments to them. And their spiracles are a little bit higher than on exotids, usually between the 3rd and 4th leg. Now, while exotid ticks will go from a larva to a nymph to an adult, the soft ticks are a bit funny. They'll go from a larva to a nymph, and then maybe to a nymph, and maybe to an adult, or they might go to a couple more nymph stages and then an adult. And it does not vary from species to species. I've reared relapsing fever ticks and had them go from a nymph to a nymph to an adult. And the same batch of ticks had some go from a nymph to 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 a nymph, then to an adult. Argassids are quick feeders. Hard ticks need to stay on that host for days, but soft ticks can usually complete their feed within about 10 minutes. But this suits them since they're most entirely adopted the nesting strategy. So these ticks will live in caves, animal dens, bird nests, where they can stay with their hosts. And when they need a meal, they'll go up when the animals are sleeping at night and get it, and then they'll get gone. So here's some rather notorious ticks. So the first one is the fowl tick, Argus persicus. So their hosts are birds, especially chickens and turkeys. They go after ducks, songbirds, and sometimes small lizards. Soft ticks are a lot less picky than hard ticks, so they'll at least try and bite in if you're warm. So they'll generally bite humans if you give them the opportunity, but this is usually only incidentally. So Argus is a vector of Egyptopinalia, palorum, and Borrelia anserina. Now, the fowl tick was notorious for parasitizing chicken houses. And they got rid of it when we moved from having wooden flooring in the chicken houses to concrete flooring in the chicken houses. When they had the wooden flooring, the fowl ticks could come up through the cracks in the wood, feed, and then dive down into those cracks to avoid being made a meal of themselves. Now, this is kind of an important tick because backyard birds are becoming a thing now, where people are keeping chickens in their backyards, and they don't have concrete flooring for their chicken houses. So this tick persists in the wild, and you should always be on the lookout for it if you're keeping backyard birds, since we probably do not want this thing back. The other notorious tick is Atobius magnani. This is a spinous ear tick. The hosts include cattle, horses, mules, sheep, goats, cats, dogs, wild canids, rabbits, elk, white-tailed deer, mountain sheep, goats. They like it all. Mostly they prefer large ungulates, especially cattle and horses, and they get up into the ears, and that's where they like to stay. Interestingly enough, the adult stage of this tick is non-feeding, and their mouth parts actually become reduced. And they sit there, persist, mating and laying eggs very slowly over time. They're not a known vector of pathogens, but they tend to leave pretty grievous wounds, and if not treated properly, those could lead to an incidental infection. The soft ticks you kind of want to be on the lookout for is Ornithodorus turricana and Ornithodorus talahe. And these are relapsing fever ticks. So both are encountered infrequently. These are nest-dwelling ticks. They'll live in caves and dens and things, but they are vectors of the causative agents for tick-borne relapsing fever. This includes several Borrelias, in fact, many of which are known for the species for which they're named. There was an outbreak a few years back, you may have heard of it, in Austin. There was a small convention for authors, and they were in a cabin near the caves where they have bats. And these ticks got out and under that cabin area and started biting the residents. And they all came down with relapsing fever. You should also be on the lookout if you ever decide to go spelunking. An associate of mine, Colonel Kim, did research on these ticks, and he would toss a small chunk of dry ice into the cave, and within moments, you would see the ticks raining down from the ceiling of the cave. So any host that happens to be walking in there, whether it's a turkey vulture, a deer, or you may find yourself getting some bites. So are ticks resistant to adverse environmental conditions? So this is a shot I took with my drone of Bryan, Texas, a day after Winter Storm Uri. Everything was white and snowy, and the ground was suffering from a hard freeze. We went down to the valley to our research site at Laguna Atascosa, and this is 2 weeks post the winter storm, where there were carcasses all over the place. The trees had been skeletonized. Everything was brown or gray and looking very, very dead. And we put down some carbon dioxide traps. What we found out is that, yeah, the ticks survived. They're highly resilient, and they can survive in very adverse climatic conditions. So within an hour, our traps were coated in ticks. So the most important way to not get bit is prevention. So know where you're going and if there may be ticks present. You want to wear light-colored, full-length clothing whenever possible, because you'll always be able to spot the ticks better. You want to cover exposed skin surfaces with a repellent. DEET or Picaridin is recommended. You want to tuck your pant legs into your socks to keep crawling ticks atop of clothing where they're visible. The better you see them, the faster you see them, the better. And you want to use spray-on clothing repellents that contain permethrin. There's a wide variety of pyrethroid toxins. Now, permethrin is not a repellent. Permethrin is an insecticide and a caricide. And I highly, highly recommend it because it kills the ticks as opposed to just repelling them. And there are some ticks, including Amblyomma americanum, the Lone Star tick, that do not care how much DEET you cover yourself in. They will come in for a bite, but they will get stopped by permethrin. The best way to use it is to spray it onto your clothing the day before, let it dry. It will subsist on that clothing for several days and be active, so you don't have to worry about reapplying it. So you want to perform frequent and thorough examinations on your clothing. Check your exposed skin for crawling or attached ticks. Don't do what my mentor did and not check between your toes if you're walking around barefoot, because they will get in there. You want to avoid wooded and brushy areas, especially places with high grass and leaf litter. Now, when I say high grass, I'm not talking about, up to your waist or something like that. think of if it's about tall enough to reach the underside of a medium-sized dog. That's what you're looking for. You want to walk into the center of the trails. You want to remove all outdoor clothing, sleeping bags, blankets when you're done. Place them directly into a dryer at high heat for at least 15 minutes. And this will kill the ticks, unless that clothing is wet, in which case you want to do about 90 minutes. So cold and medium heat will not kill the ticks. Remember, these things are very good at surviving in adverse climatic conditions. Tick removal. So here's a nymph that's feeding on me. This is Amblyomma. It's got its mouth parts inserted, its palps are split to the side. So what you want to do is take some tweezers or tick spoon if you have one, grasp as close to the head of the tick as possible. And then you want to use a slow, steady force to pull that tick away from the body. Remember that that tick is anchored by that toothed hypostome and superglued in by its saliva. But by pulling very slowly and avoiding quick jerks, you should be able to extract the tick entirely. Now, if the head does pop off and remain in the bite, leave it alone. Please do not use a razor blade to try and excise it or use pins, or there's no need to run to the doctor either. Just treat the bite with alcohol and soap and water and leave it alone. Now, the removed tick can be placed in alcohol, or the USDA method is to place it into a plastic bag, seal it up, and put it in the freezer. Now, why do you want to do this? Well, one, it's going to kill the tick. That's something everybody wants, right? The other reason you want to do this is because you want to have that tick handy in case you start showing symptoms of something. So if you're starting to feel ill after a day or two, you'll want to take that with you, bring it to your doctor. The doctor can identify that tick or send it off for identification and figure out what might be causing you grief. So now, I've been told many, many methods of tick removal, but I beg of you, do not squeeze the tick, burn the tick, cover it with petroleum jelly or varnish, coat the tick with beer or salt or urine, and please do not attempt to excise it with bladed instruments. So, you live in Texas, so you get to take advantage of a remarkable tool. Texas A&M AgriLife Extension has the Tick app. It isn't actually an app, it's a website. You can access it through this scan code or directly through the my mind is blanking. What is this? The address there. Now, this has information on AgriLife Extension services, which includes pathogen testing. So they'll be able to take the tick that's bitten you and test it to figure out what harmful things it's got in it. They'll be able to identify the tick, and it also includes things like preventative measures and caution about exotic ticks. Do we have any questions? Hello?
Go ahead and ask.
Okay, my question is, what is the benefit to it tick being a multi-host tick? Like, if it already has a good host, why would it fall up and wait to find another?
Well, there's a couple advantages, and there's also the fact that ticks are influenced by the pathogens they carry. So it may be easier to get onto a different host at a different life stage. If you're a one-host tick, well, you've got your meal there all the time, but you're also at greater risk of being groomed off by that animal. Whereas if you're a multi-host tick, you can get your meal, drop off, and then find another host, which reduces the chances that you're going to be groomed off.
Okay. I have a question. What in the world are ticks good for? Does anything eat them? Do they do anything good for the environment or for animals?
Well, they're certainly good for the pathogens they vector. There's a lot of apes that eat ticks, though this is kind of incidental. There's a couple different birds that actually specialize on ectoparasites on big animals, but that's mostly in Africa.
So do they have any natural predators in the valley?
Not really. There's a lot of things that will opportunistically go after a tick. There's talk about fire ants eating them incidentally. Talk about crabs that will go after them incidentally. Many animals that groom them off will in fact eat them. In fact, this is a way that some dogs can pick up things like canine hepatozoonosis.
What is that?
It's a malady that infects canines. It's caused by a bacterium.
Did Judy have a question in the chat?
I'm going to read some chat questions. Where's the best place to find the best incidence data on tick-borne diseases in South Texas? Do medical doctors have mandatory report?
There's not mandatory reporting for most doctors, and I've run into several doctors who will claim up and down that they don't have ticks and it's not a problem. Your best bet is to check up on the tick app from Texas A&M AgriLife Extension. It's got all the information you need on where the ticks are and things you should be looking out for.
Do ticks drop out of trees? And if they do, are they just coming from a bird's nest or where?
So I have heard of the mysteries of the tick rain, and I'm told by many of my professors that it does not happen, apparently except in Oklahoma, where they do have tick rain. So I suppose as long as you're not in the forest of Oklahoma, you should be safe. Ticks that are questing are only going to get up as high as about a medium-sized dog.
So would there be any chance that they would fall out of a bird's nest and get on another?
You might potentially get a soft tick that might fall out. But again, that would be incidental. The closest thing to tick rain you'll find is walking into a cave. Spoiler alert, do not go into the caves in Government Canyon. This is a bad idea.
Another chat question. What kind of symptoms did you watch out for after a bite to know when you need to go to the doctor?
Well, muscle weakness, general malaise, fever. Of course, with Borrelia burgdorferi, the causative agent for Lyme, you want to look out for that bullseye rash. Now, that bullseye rash will also appear for Borrelia lawrenceii, or southern tick-acquired rash illness. But in either case, you probably want to see a doctor and get a treatment with some doxycycline. Generally, if you are feeling unwell and you know you have bitten by a tick recently, it's probably a good idea to go and see your local doctor. Now, as far as Lyme is concerned, the bacterium is not immediately transmitted. So if you're bitten by a black-legged tick, you have anywhere from 24 to 48 hours post-bite where that bacterium will be likely transmitted to you. So if you get it off immediately, your chances of not getting that increase immensely. Now, this is not true for viruses. Unfortunately, things like tick-borne encephalitis, heartland virus, bourbon virus, Powson virus, if that tick is attached to you for about 10 minutes, there's a good chance that if it's infected with that virus, that you've got it.
How long does it, like, once they're bitten on a host, how long does it take for them to get a sufficient meal to drop off and to go to the next life cycle?
So it depends
Or
on a
like
couple things. One, it depends on the species of the tick. Ticks grow at different rates depending on their species. And also it depends on the host. The tick has an immense pharmacological arsenal contained within its salivary glands. These act like skeleton keys to your immune system, since they don't know that they're getting up on you, especially the generalists like Lone Star tick. They may be getting up on a coyote, or may be getting up on a chicken, or you. But it's got the tools it needs to defeat your immune system and keep that feeding lesion open. But some hosts are better at closing that feeding lesion than others. So the tick, once it's attached, has to play the game of trying to fight off your immune system in order to get the blood it needs to survive. If it's losing that battle, it may take it a little longer.
So then if it doesn't finish it, and let's say it gets knocked off, it would just find another host?
So no, actually. After a tick has started blood feeding, there's upregulation in its genes going on, where it switches over to feed mode. So once that tick starts feeding, if you pop it off, it is not going to find a new host.
I have a question. As the tick attaches, just before that, where is it? Is it on the ground? Is it on a leaf? Is it on a little branch? Where is it?
Well, most ticks quest on grasses, but they'll also get up on small branches, usually, like I said, no taller than about the underside of a medium dog. And as you brush up against it, it'll reach out with its legs, grasp onto you, and then immediately start climbing and testing you with its palps to see if you're a good host.
So is it jumping?
Ticks are incapable of jumping. Their legs are cursorial only, so they can only walk or run in some cases.
It's funny because I'm a gardener, so I'm always in the garden and in the soil and everything. I've never seen a tick, never. Maybe I'm not
Well,
looking.
a lot of ticks are specialized. So the ticks I've discussed, they might go after people, or they'll definitely go after people. But that's not all of the ticks that are in Texas. There's a large number of ticks, but they're specialized in other things. So they'll only go after rabbits or rats, or they'll only go after pigs or cattle.
I see.
And remember, in general, when we think about parasites, the parasitism rate for any given population is only about 20%. So most of the hosts you're going to look at are probably going to be relatively free of parasites.
Where do they like to lay the eggs? On a
So
host or on the grass or whatever?
they need to lay their eggs under leaf litter, in the ground. They'll often go to the grass node where it's coming out of the ground and crawl down into that under the ground. The eggs are susceptible to ultraviolet light, so full exposure to the sun is going to kill them, just like vampires. So The female will crawl off into the environment and sequester herself away in the shade, away from other things that might get at it. And then she'll start laying her eggs, which can be anywhere from 1,000 to 50,000.
I have a question. So I do work in the brush and maybe about 30 minutes to an hour prior to going into the brush, I do spray, from like head to toe. And I spend about maybe 4 to 5 hours in the brush. Is there a reapply, do I need to reapply, like after so much time, or is like one good application fine?
I would look at the label for whichever product you're using. Not sure what it is for DEET or Picardin. I know that for permethrin, as long as you've left it to dry, it's not going to, wash off immediately if you get hit by rain or if you sweat a lot, and that will last on that bit of clothing for several days. An entomologist trick I learned was, in a pinch, if you don't have permethrin, but you do have bug spray that has some sort of pyrethroid in it, you take the clothes you're going to wear for the next day, you spray them, and you let it dry. Once it's dry, it should be safe for you to wear, and it will keep the ticks off. But I highly recommend going into, say, a outdoors type store like Bass Pro or a hunter shop where they'll sell actual cans of permethrin.
Thank you.
Is Picardin or DEET any better, or they're basically the same?
They have different efficacies against different ticks. In general, they're pretty good, and they'll also help keep you free from things like mosquitoes and chiggers. But like I said, there's some ticks that they do not care. Amblyomma maculatum, the Gulf Coast tick, and the Lone Star tick don't care if you're swimming in DEET. They will come and take a bite.
So on those, Picardin might be better, or they don't carry that either.
They don't really care about that one either. I mean, you're generally going to keep a lot of ticks off, which is the point. The best way to keep ticks off of you is to be conscious of your environment. Are you walking through areas where there may be ticks? Are you keeping an eye out for ticks that may be crawling on you? Keeping in mind the fact that their larvae are very, very small. So don't just be looking for big things. Look for things about the size of a period that are moving up on you.
Any other questions?
That's like 20 minutes.
I know, very interesting. Well, thank you so much, Brian.
Great presentation.
You're quite welcome. Thank you for letting me come out and speak to y'all.
Well, thank you so much. I learned a lot.
This is really interesting.
Yes.
Great, well, that got really great.
Well, thanks again, Brian, and you're welcome to stick around on WebEx for our business meeting if you really want to, but I know it's an hour later on the East Coast, so we appreciate you taking the time to be with us.
Like I said, thank you very much for having me, and y'all have a very nice night.
Thank you.
Thank you. You'll be scratching off.
And that concludes our presentation, and we'll go ahead and take maybe a 10-minute break, and then we'll start our business meeting. All right, it's 1:45. Let's get started. First up, we have the treasurer's report from Gail. Yeah, it's fine. Okay, not much to report.
kind of a slow time, right, of the year. One thing we did do is we paid our $500 sponsorship toward the state meeting that we had agreed to do. And so we did take care of that this month, and looking forward to that state meeting upcoming.
All right. And next up is Velma with the minutes from the last meeting. Is there any updates or changes? All right, the next up would be the second vice president, Jennifer.
Hi, everyone. So I am working on lining up presentations for August and September. We do have October, so we had Brian Rich tonight on ticks, and we have Bill Rich in October on gardening for butterflies. And Bill helped volunteer to do that program, and also got Brian to talk to us tonight about ticks. I really didn't know anything about ticks. I learned so much. So if you have any ideas for presentations, please let me know. I have a few leads, but I would like learning about everything. So come talk to me if you have an idea.
And next up is Jaime with the membership committee.
Afternoon or evening, everyone. So the report here has been updated. One of the things that I've done is I've gone back and members reporting and different opportunities have been updated according to the numbers that we've seen since June. The other thing also is adults served and youth served. These are numbers that Tony Ressinger has requested to be in the reports, as this is what he's reporting to Texas A&M. Okay, so other than that, the July number just reflects what we've been doing up to this point in time. What has changed, though, is down here at the bottom, the value had been reported at $28. It is actually at $33.49. So the values here have changed. Okay, so that's actually been updated. And as things change, I'll keep you guys abreast of that information.
All right, so next would be their awards and recognitions. For the initial certification is Melissa DePagter. Is she here? I hope I said your last name. You did. Thank you. You're very welcome. And also would be Chantel Ortiz. And for the 2024 recertification is Lisa Adams. A 100-hour milestone is Stephen Blam, Bing, and a 500-hour milestone, Ellie Kidd and Velma. Come on down. Oh. And congratulations to those people that are actually online with us. Good job. The next item up would be Anita. She's not here.
Yeah, it's online. She didn't send me anything, so she may not have it.
Was that for me?
Yeah, you have.
Yes.
Oh, I just want to mention that on Saturday from 2 to 4, I will be doing a creative workshop at the creative photography workshop at the McAllen Library. If anyone is interested, that would be 2 hours of AT. And then in the evening at Quinta Matzalan, they are kicking off Moth Week from dusk to midnight, and they will have some stations of mothing. And that's, of course, an open invitation to anyone that would like to join us to help volunteer. And don't be put off about volunteering if you don't know anything about moths, because somebody will be there to help you, but it would be a good opportunity to learn something new. And that's all I have. Thank you.
Thank you. And next up would be Judy.
Judy's online, but she's not feeling good, so she had asked me to read her stuff. Oh, yeah. Okay, Anita mentioned her creative photography workshop and mothing by moonlight at Quinta, both on the 20th. The Quinta mothing is going to go from 9 p.m. till midnight. The same night, Estero State Park is doing their night hike with mothing. They'll probably walk around some, but it's going to have a focus on around their moth sheet setup. That'll be from 8 p.m. to 10 p.m. Benson State Park is doing their moth night on the next weekend on Saturday, July 27th from 8:30 to 10 p.m. That's the same night we're doing our chapter field trip moth night at Oleander Acres. We'll start at 9 p.m. and go till midnight. So if you wanted to do both, you could check out Benson, then come over and check out us. Let's see what else. Kim, do you want to talk about your?
Yeah, sure.
Yeah.
Okay, so if you're interested in learning about fungus, there's going to be a presentation called Our Friend the Fungus, and it will be this Wednesday on July the 17th from 6:30 to 7:30. It is in the beginning before the start of the Master Gardeners meeting, but you don't have to attend the meeting, just go for the presentation, and it's before the meeting. So it will be from 6:30 to 7:30. Oh, it's in San Juan. So it is in the location, I don't know if you're able to see it there. It's 509 East Early, and it's in the multi-purpose building. It's not at the garden place. It's at the building there. There's going to be a sign. You just park in the back, and the door will be propped open. Mhm.
I forgot to mention this is Moth Week, the July 20th to the 28th. You can participate in that on iNaturalist, and that counts as if you're learning, come do a presentation, that counts as AT, and if you're doing citizen science, that counts as volunteer time.
All right, up next will be River.
Hello, everyone. Okay, so first I want to mention that the awards part, some of you are very close to recertifying. I want to encourage you to watch any TMN Tuesday presentations that you have not watched yet, so that you can get enough AT hours. And there's a few of you that just need 2 or 3 or 5 volunteer hours to reach recertification. So get on that, please. All right, for volunteer opportunities, we have our ongoing ones. We have our chapter projects that are the Vanny Cook, the Pollinator Garden at St. George's, which is here, and the Sherry Heights Butterfly Garden in Mission. So those are opportunities that you can contact the people that are in charge of them. For Vanny Cook, it's Becky Jones. For the garden here at St. George's is Robert. Hernandez, and for the Sherry Heights Butterfly Garden is Logan Dovalina. And I will be sending out their contact information very soon. The other ongoing ones are nature centers here in the valley, including the state parks, McAllen Nature Center, Valley Nature Center, Frontera Audubon, Quinta Mazatlan, all those places, Santa Ana, things like that. There's this little pamphlet that I put in the back as you leave. If you don't have it, pick one up. It's got a lot of them in here that you may be interested in going to. We also have citizen science, the Adopt-A-Loop, and Donna has a little presentation she's put together. You want to come up and?
This is just a quick reminder that we do have an approved chapter project, the Adopt-A-Loop project, and Ellie Kidd is helping me, and we kind of co-coordinators the project because we have two different sites. You want to go to the next slide? We are here in the lower Texas coast of the Great Texas Coastal Birding Trail, and all these in green are the different sites within our service area. Most of them are along the coast and out towards the west more. Next. Okay, so if you want to do some Adopt-A-Loop observations, you'll need to take a picture of this slide. Back in 2021, during COVID, we adopted a Sterolana grande loop, and in that loop, a loop is a little trail that connects these sites. We have these 4 sites, Sterolana grande, Mercedes Civic Center, Valley Nature Center, and Frontera Audubon. When you go in to document that you're doing a naturalist for Adopt-A-Loop, you will need to know these numbers because you have to put in not only the site number, but the site name when you go to enter your observations. Next one. Now, earlier this year, Ellie saw my presentation in January, and she identified a group that were interested in doing one on this other side of the valley. And so they are going to these 5 sites. And I do want to make note of the Chihuahua Woods is now called the Pixie Preserve, and it's part of the National Butterfly Center. And that is restricted use. You have to go in with special appointments. So we're not really working in that area at this time. So if anybody of you on this side of the valley are interested in doing Adopt-A-Loop, there's some more in this area. Okay, next. And the reason we go to do these site visits is to document the wildlife, plants, butterfly, birds, anything that's out there to help entice people and encourage people to come and see what we're identifying out there. You can, we try and go on a quarterly basis, and I'm doing this because this is the beginning of another quarter starting in July. So anytime July, August, September, we encourage you to go out to any of these sites. You can do e-birding. If you do your e-birding, all you have to do is enter your data in eBird as to what you observe. The Texas Parks and Wildlife people will automatically get that information that they can upload. Also, if you're doing birding, you can, if you go in to get your hot spots, a lot of times the hot spots are the lower Texas coastal site numbers. So if you look for the hot spot at, say, Estero, it'll have that Estero site number already loaded up. So you'll know you've got the right location. Now you can do the birding with birding groups or with your family. You can do it anytime you want to. The heat is early, and hopefully you'll still see things. But yeah, we encourage you to do that on e-birding. If you go to the next slide, I'll talk about the iNaturalist observations. iNaturalist observations are not automatically picked up by the project. Some projects, they'll go and collect the information on their own. This Great Texas Wildlife Adopt-A-Loop does not do that. So when you go in and start your observations, you have to identify the project by the Great Texas Wildlife Trail Adopt a Loop project. And I'll give you another slide here in a minute. So there's a little drop down menu that makes it real easy to do. You should be able to do it without any too much trouble. You can either do it as you're doing. What I'll do a lot of times is not use up my battery on my phone so much. I will take pictures, and then after I've done all my observations, then I'll go in and put it all into the Adopt a Loop thing. That way you're not doing it for each and every observation you make. Okay, next. And here's how I do that. You can see there's the edit button. And when you go in to your observation, you hit the edit button and then next. And then you pull up projects and GTWT, Great Texas Wildlife Trails is what you're looking for. Next. And then it'll come up, it'll want the place of observation, which is the site number. And for us, that LTC is Lower Texas Coastal Loops that are down here. So all of our numbers will start with LTC. Next. So to report your hours for doing this, you'll have a VMS entry. It's going to be under FR, which is citizen science. And you just indicate whether you're doing e-birding or iNaturalist for the Great Texas Wildlife Trails Adopt a Loop project. And again, say the site name and number. That way we can verify if we need to. But keep in mind, if it's at either Estero or Benson, these two are state parks. So you go to the state park listing and then under the research, citizen science research, and then do the same information for that entry. Okay, next. And at this point, the state is not able to collect all that information. One of the reasons is because we first started these observations, as you might remember, back in 2021 during COVID. At that time, it was offered as a virtual volunteer fair project. And so a lot of people entered information on their Adopt a Loop observations under that category. And since then, they've offered it at annual meetings. And so different chapters have utilized different methods for recording their information. So the state has had a terrible time, or the Parks and Wildlife have had a hard time collecting the information from our different sites. So we're going to hope that when they redo our VMS system before the end of the year, we'll come up with a better way of doing it. So if you can let me know by email, if you do go out and do an observation, whether it's eBird or iNaturalist, and just where it was. I don't need to know the time or anything. Just if you do it, that way she has a spreadsheet for us at the state level. We've got a spreadsheet that we put the visitation dates and places on. And that's just so they can verify without having to go through ever many chapters, how many observers. You know, that'd be a really big job for a statewide effort at that, but much less for us at the chapter level. Okay. Any questions? Yes. Yes, I think we, and matter of fact, somebody else was saying maybe we could adopt another loop. So maybe we'll look at that. We've got one on the east and one on the west, and maybe we'll find something a little more central that's still not too out of range for some people to go to. And if you've got a loop in your area that you'd be interested in starting to make observations at, let me know, or let somebody on the board know, and we will officially adopt that loop. Because that is one thing we do have to let the state know, is when we officially adopt a loop. Because there's initial assessment, and then there's follow-up assessments in addition to the observations we make. Okay, and if you're going to Moth Night or some of these other things and you don't need advanced training, any of those pictures and things you do at birding trips, Moth Night, any of those, you can count those observations. If they're at one of those sites, you can count those observations for under the Adopt a Loop program. Yes?
We post everything on iTunes, so that's a problem.
No.
We may form a production on it, in case anybody interested.
Yeah, sure.
You don't just, you picture it as a whole shot. The, one of the things, yeah. On my computer. And it's necessary, they. And then, right. Anyway, basically, gives us a lot more volunteer hours.
That's very good point. All this other. Like the picture. Download it on their. And do a lot more editing and then batch. One more page in there. One at a time. Batch process what's in there.
And I like to try to, identification. Probably most of the time, but not always.
It's valuable to do that cropping up stuff on your computer or even if your phone, if you're on the phone, because you post a picture of a bird and the bird takes up 10% of the photo, no one's gonna ID that. They're just gonna go right past it.
Anything else? All right. Another citizen science opportunity is the red crown parrot count. It's done quarterly, and we get an email about it each time it's time to do it. So keep an eye out for that email. It usually comes from Javier de Leon. So on Saturday the 20th, Saturday's coming up, we have Fairy Fest at Edinburgh Scenic Wetlands from 6 to 10 p.m. The chapter is going to have a booth there, and that's a volunteer opportunity. If you want to help out on that, you can contact me. I will be there. I already have three people that said they're going to go. Also, on that opportunity, Melissa and Melissa, created a fairy house because we entered the fairy house contest that they're going to have. So that's part of our promoting the chapter is to enter that contest. So if you have a chance to go as a participant, if you're not going to go as a volunteer, you can go by and take a look at the fairy houses that are going to happen. Ours will be there. Yes. Yes, we'll take pictures and we'll send them to Joseph, and he might put one or two in the. Oh, okay. Well, yeah, I don't do Facebook, but maybe somebody else does. I don't post. We are sort of a little bit. We are. Oh, absolutely. Absolutely. Yeah, and Kimberly was very creative and made a bingo activity for the kiddos. She's got it here. If y'all want to look at it, it's very interesting. And on the table in the back also, for October, Rio Reforestation. There's a flyer back there. You can take a picture of it. It has all the information. That's another volunteer opportunity. I think there's going to be a training. There is going to be a training for any of us that want to be leaders, that will manage a group of people digging holes for the trees that we're going to be planting. So if you're interested in that, please let me know so that I can pass that information on to them. And as far as I know, that's it on the volunteer opportunities.
Okay, next up would be education director, but Jim is not here, and I noticed Roxanne is not also. Is she online? Yeah, okay. Next up would be Kathy. Thanks, Roberta. Well, you all heard about how important it is to volunteer and report your hours. And even if you only do 15 minutes, please write it down as 0.25 and River and I will be happy to approve it. Every little bit helps. But the one thing I want to point out is we've got a fantastic opportunity coming up in October with the state convention. And the first week of August is when the sign up begins. This year, they're going to offer a total of 142 different classroom sessions. Unbelievable. So you name it, from turtles to insects to mothing, it's all there. Plus there'll be another 32 field trips that actually start the day before. So that's October 24th to the 27th. It's in San Marcos, so it's only about a 4-hour drive. They are offering hotel at the Embassy Suites. And if you have 3 people to a room, it turns out to be a total of about $72 total per person for the 3 nights. Not too bad. There's other places nearby too, and that does include your breakfast and parking. The price seems a little steep, but we'll be talking with our president and the board, because in the past, they've always done some type of a reimbursement for people that sign up. And I know that the cost of $450 seems high, but I noticed that if someone wants to just come and attend meals only, of that $450, $375 represents the food cost, because they're including some fabulous meals over the 3 days. So that's part of why it's high. Last but not least, I just want to say I've been to only one state convention, and it was fabulous. The people you meet, the opportunities you do. They also have an art contest where you can take a picture, sculpt something, make a quilt, and you enter it by actually taking a photograph. So it's a digital entry. So any of you that might be a little arty, think about that too. And I know we'll also have perhaps a chapter project, and we also do a nomination for Advisor of the Year, which I know Donna's been working on with Tony. So if there's any possibility you could carpool or get together with folks towards the end of October, give it some serious thought and watch your email, because the sign up will begin sometime in the first week of August. So that's all I have to say, and we'll try to get this wrapped up so you get your half hour of 0.50 of volunteer time for the meeting. Here you go, Roberta.
Kelly, are you online? Yeah, she's online. Do you have anything to add, Ellie?
Are you guys talking to me?
Do you have anything to report?
Yes, I do. I got an email today from Texas Parks and Wildlife. They've received our bat data that was collected. Thank you to River and Donna and Kathy for getting those monitors set up. And they should be getting the data back to us. And next week is training for the analysts, which I just sent an email out today about. And so we're on a roll. Hopefully we got a lot of bats out there. That's it.
Thank you. Everybody might not be aware of it, but Tony Reisinger is going to be retiring at the end of the month. And he's been our advisor, I don't know how many years. I know he was put in for advisor award back in 2013. So we want to be sure everybody's aware of that. And so you can give him congratulations, and we'll have him in for a special evening and cake and something here before too long, hopefully. All right, that should end our meeting tonight. I just want to make an announcement that the next board meeting is August the 5th, and the next general meeting will be the 19th. Does anybody have anything else?
I just want to clarify that it's an hour of 18 for the presentation, and then half an hour for the meeting plus your drive time. Right?
Correct. Good night.
Society being a lot of years, they're also, I did that last year.